I'm Rafael Amado. I'm the head of research and development at Adaptimmune. I'm a medical oncologist, and I also serve as the chief medical officer for the company. So we work on T-cell receptors, affinity mature T-cell receptors against antigens that are only in cancer cells. The data that we would like to highlight is data with an antigen called MAGE-A4. We have a pilot trial that is now in the expansion phase, and we've treated a number of patients with a variety of tumor types. One of those is synovial sarcoma, because we proved that MAGE-A4 is expressed at high level in synovial sarcoma. We've treated a total of 10 patients to date, and we've been incredibly impressed by the responses to date. So 5 patients have actually had responses. One of them is still to be confirmed. And the rest of the patients, except for 2, have had dramatic shrinkage in some of the largest dimension of the tumor. And it's possible that with further CAT scans, we will see these patients turning into a partial response. These are patients that have exhausted all therapies. And so this is actually quite important for the sarcoma field, where immunotherapy actually doesn't have any activity. In the non-sarcoma patients, we have seen sporadic evidence of activity. We had a patient with partial response in lung cancer. We had a patient with a mixed response in both ovarian cancer and melanoma, where liver metastases have shrunk. So we think that in epithelial tumors, we still have a bit of work to do, but we're working on those techniques in terms of making the cells more potent and making the vector also be able to target the cells, including the tumor microenvironment. Immediate thing that we're gearing up to doing is to launch a phase 2 study in synovial sarcoma and myxoid round cell liposarcoma. That's another tumor that expresses MAGE-A4 at high level. And this would be a single-arm study with about 60 patients. And we're looking for a response rate as the primary endpoint, as well as durability of the responses. So this study is supposed to commence this year, and we will be treating patients before the end of the year. In addition to that, we have developed a new version of our MAGE-A4 TCR that contains a CD8 alpha, and the reason for that is to engage CD4 cells to actually kill tumor cells. Normally CD8 cells are the cells that are tumoricidal, but CD4 cells lack the CD8 receptor, so we're engineering the CD8 receptor into the CD4 cells, and we've shown preclinically that they're able to kill tumor much more potently than those cells that don't contain CD8. And in addition to that, they can engage antigen-presenting cells, and they can spread the autoimmune reaction by engaging other antigens beyond MAGE-A4. That study is called SURPASS. The study that is the phase 2 study in sarcoma is called SPEARHEAD. So both SPEARHEAD and SURPASS are supposed to launch this year. The SURPASS IND was just opened about a week ago, and we are opening sites as we speak. So we're really excited about both the first generation and the second generation of our MAGE-A4 TCRs. We have other TCRs in the clinic, and obviously a lot of TCRs in discovery that are earlier, and they may be INDs in the future. One of the TCRs that's in the clinic that's moving through dose escalation is against alpha-fetoprotein in liver cancer. We're still at the 1 billion cells, so we're still relatively early, but we've seen promising drops in alpha-fetoprotein levels, as well as shrinkage of tumors and metastases in patients, even at lower doses. We think that the peptide HLA complex is the right one. There's another company that has seen also a similar benefit with another product, which is a TCR mimic, and we're targeting the same complex. So we're very excited about what will happen once we get into higher doses. And then we're very focused on allogeneic products. We want a homogeneous product that has the highest possible potency, and we have a program that is increasing in size and resources that derives T cells from iPSC, or pluripotent stem cells. That program is moving along quite nicely in the company. We're able to get T cells out of iPSCs, and those T cells are functional. And we're working with our collaborator Universal Cells, which is a company now of Astellas, to do the genetic editing part to to make the cells invisible to the immune system. So we have a portfolio of TCRs, and also we have a platform to put these TCRs into allogeneic cells in the future. And so, and in addition to that, we're on our way to getting the first TCR approved against MAGE4 in synovial sarcoma. So these are great days for Adaptimmune.